A very, very heartfelt welcome to everybody here. It's Johns Hopkins Medicine time, winter time, we're here. As a matter of fact, this is a momentous occasion because you'll see something about a 25th anniversary, and that's true. It's 25 years that we have been coming to South Florida to share with you the exciting discoveries at Johns Hopkins Medicine. Now, I have a question. Who here was here 25 years ago? Is there anyone? Oh my God, Mike. All right, that's wonderful. Wonderful. Now, Nancy and I lay claim to that. And we'll take it one step further and say that we have been here every year. Now, who has been here every year? Mark, that's absolutely. So anyway, we let these other academic medical centers now kind of compete on our turf, because I think you're all sort of maybe exhausted by all the events taking place from everybody coming to South Florida. But remember, we were number one. All right, we're the granddaddies, and we try to be number one in everything that we do. So that gives me the occasion to introduce to you the president of Johns Hopkins University, and I must say that in the 25 plus years that I have served as a trustee and as chairman of the board, this has been a glorious period in Johns Hopkins history. Every academic medical center, every academic division of Johns Hopkins University is absolutely thriving, pulsating. It's a glorious time in our history. And our 14th president is the person responsible. He's the one who's giving it that pace, that sense of urgency, meaning that we want to do things well. We want to be the first in what we're doing. And wonderful academic leadership throughout the entire university. We are extremely privileged in that regard. And the person responsible is Ron Daniels, the president of Johns Hopkins University.
Well, thank you so much, Morris, for that very kind introduction. But look, again, on behalf of all of us, to you and Nancy, we salute you. We salute your imagination, your vision, that you brought this symposium together 25 years ago. And I checked, Morris, coming in, confirmed with Steve Rum and with others, that 25 years, year in, year out, no shutdowns at any point in time. This symposium has continued and has flourished, and it constitutes a wonderful touch base for the university and the School of Medicine and the health system with our great friends here in Palm Beach. And so again, to Morris and Nancy, thank you. Thank you for so much that you do, but thank you for this great act of leadership. Please join me. So, as Morris said, with no hyperbole whatsoever, this has been an extraordinary, indeed glorious time, as he said, for the university. Last November, Johns Hopkins, as you may have heard, received the largest philanthropic gift in the history of higher education, $1.8 billion for our financial aid endowment. I introduced Mike Bloomberg a few months ago in Washington, an event where we were celebrating the first anniversary of our Bloomberg American Health Initiative, which he established a few years ago with a $300 million gift. And I said, that was $300 million gift. That used to be a big gift at Johns Hopkins. But now, of course, with this gift, he has yet stretched our imagination and our sense of possibility quite dramatically. This dramatic infusion of funds will allow Johns Hopkins to join the pantheon of truly great universities that are need blind and no loan for undergraduate students. The gift registers a powerful, a stirring affirmation of the importance of ensuring that the most meritorious students are able to participate fully in a Johns Hopkins education, irrespective of their family's financial circumstances. And to me, this represents the great promise of the Jeffersonian ideal of equal opportunity that lies at the bedrock of America. And so when we're in a time when we can't agree on many things, at least we can agree on that. So that's our first bit of great news. 2 weeks ago, you might have seen that Johns Hopkins Washington, D.C. programs will be moving to an iconic new state-of-the-art home in the landmark museum building located on Pennsylvania Avenue, a short hop and a step from Capitol Hill, and strategically located right next to the Canadian Embassy. When we made the announcement 2 weeks ago, my hometown, my home country. When we made the announcement a few weeks ago, I got a call that day from the Canadian ambassador who told me essentially, there goes the neighborhood. I said to him, Ambassador, bear with me. I've got a great idea for how we can really get the synergies maximized between our two buildings and our two countries. And that is, I think we should burrow a hole between the two buildings and put a Tim Hortons Donuts in there to service us all. He was still not entirely convinced. But every part of our campus, every part of our university will have an opportunity, will be invited to be participants in this building. Paul and I have already scoped out the office that we're going to share and where we're going to sit and work together in our new digs in Washington, that not moving there full time, but intend to be able to participate in that building. And finally, our SNF Agora Institute, $150 million interdisciplinary career interdisciplinary center, recently founded to study and address the challenges of polarization and discord confronting democracies around the globe, is actively recruiting its founding faculty and commencing work on a Renzo piano designed building in Baltimore that will house its activities. Just last week, PBS's Judy Woodruff hosted an Agora discussion with the sitting governors. It was really quite wonderful, of New Hampshire, Pennsylvania, and Maryland, that focused on how each of these men navigated the challenges of governing with legislatures controlled by the opposite political party. So if there's hope for America, it's in many of these states where truly these branches of government have to collaborate and have collaborated effectively with one another. Of course, as important as these moments are, none of them materialized out of thin air. They are the culmination of dedicated work matched by fearless vision by a remarkable cadre of faculty, staff, students, and of course supporters. Indeed, this combination of imagination and daring do has, since our founding, defined the essence of the university and of course, its celebrated School of Medicine and health system. And it is especially evident in our pursuit of discovery fueled by basic, curiosity-driven research that you'll be hearing much more about today. So consider one poignant story. In 1916, a young medical student, Jay McLean, made his way to Johns Hopkins. His ambition and creativity were immediately spotted by Dr. William Howell, the founder of the school's Department of Physiology. Howell had a lingering question to pursue in his research on blood clotting, so he set Jay McLean loose on the problem. After months of experimentation and study with a break in between to tend wounded soldiers in the First World War, McLean discovered a powerful blood thinner. Howell would later name it heparin. You, of course, will have heard of it. Today, as you know, heparin is one of the most widely used treatments to prevent the blood clotting that can cause heart attacks and strokes, and is a fixture on the World Health Organization's list of essential medicines. In the century since this discovery, we have continued to support and inspire the kinds of imaginative and dedicated researchers whose therapies bring life-saving treatments and therapies into people's lives that change their lives and their sense of possibility. And nowhere is this more evident than in the work that is undertaken at our Bloomberg Kimmel Institute for Cancer Immunotherapy. This year alone, our researchers and clinicians proved that two of the groundbreaking cancer immunotherapy drugs developed from our research, Keytruda and Opdivo, significantly increase, in some cases up to 50% the long-term survival odds of patients with certain cancers, including melanoma. And the BKI is, of course, not alone in advancing a simply staggering array of breakthroughs. At the Institute for Basic Biomedical Sciences, scientists collaborate with clinicians in fields ranging from epigenetics to obesity to metabolism to cell dynamics. And through Bluefield Innovations, a collaboration with investment management firm Deerfield Management, we're supporting early-stage research with the potential to transform fields. And Dr. Mariki Leho will soon describe one such transformation. Indeed, her investigations into how interference with certain cellular pathway may kill cancer cells without harming healthy cells could revolutionize cancer research yet again. From Baltimore to Washington, from our founders, and Jay McLean, to the extraordinary clinician scientists and entrepreneurs across our university, Johns Hopkins has been redefining what it means to be a pioneering research university devoted to discovery and to humanity for more than 140 years. And we are fortunate, truly blessed, to have among our leaders Dr. Paul Rothman, himself a clinician scientist, someone who plays all ends of the keyboard, who knows good clinical care, but also knows good bench science. He himself has research that has been very important in illuminating the study of cytokines, and it's yielded insights both to the causes of leukemia and common allergies. In this respect, Paul is someone who understands all aspects of our mission, is a perfect leader for this moment. We're so blessed, as I said before, to have him as the Dean CEO of Johns Hopkins Medicine, and it's my great privilege to introduce him to you. Thank you again for being here with you, and we look forward to celebrating our 25th anniversary with you. Paul.
Good afternoon. Thank you all for coming today. I will say, as I'm thinking through the opportunities in Washington, D.C., our preventative cardiology program is looking forward to the Tim Horton donut shop. I really wanted to thank everyone for coming out today and first introduce, we have, our trustees are not paid, and they really do this as a volunteer to help Hopkins be successful. We have several of them in the audience today, and I'd like to take the opportunity to ask them to stand so we can thank them. Jeff Brown, Davis Camillier, Harvey Eisen, Jay Lenro, Gilbert Levin, Mickey Miller, Morris Offit, Chris Poindexter, Mark Rubenstein, Mel Shattuck, and Howard Rowley. Could you stand up so we can give you a big round of applause? Thank you again for coming and being with us today. We're trying something different this year. So we have what's called a graphic recorder over there who is going to record what he hears today, and at the end we'll have a summary of all the talks in a graphic. So thank you so much for joining us today. What I thought, for those who may not know Hopkins, just give you a little summary of Hopkins Medicine and who we are and who we've been. We are an $8.2 billion enterprise this year. That is a combination of our School of Medicine, our hospitals, our ambulatory centers. We have 6 hospitals, 2 in Baltimore, 2 in Washington, 1 in Howard County, and 1 in St. Petersburg. We have 4 large ambulatory centers, 40 primary care sites. We have 41,000 employees. We have about 1,200 medical students and doctoral students. Hopkins University has had 20 Nobel laureates come through its gate, and Hopkins Medicine this year will have just about $900 million of external research funding to fuel the innovation engine that we're going to talk about today. We also either consult or manage health systems in 15 countries around the world, from Beirut, Istanbul, Peru, Chile, Panama, Saudi Arabia, UAE, China, Delhi, India. So we help people around the world try to provide Hopkins level of care, and we're very proud of our impact around the world. One of the big changes this year was that Ron Peterson retired as our health system president, and we recruited Kevin Sowers really just about a year ago. Kevin came to us from Duke University, where he was the president of the Duke Hospital for 7 years. Prior to that, Kevin trained as an oncology nurse, and he's been just an outstanding leader with true vision about building an integrated healthcare delivery system that provides the safest and highest quality care, has already made a tremendous impact on the institution. He is sorry he can't be here today. He is in St. Petersburg trying to fix some problems we are having at All Children's Hospital, and he gives his apologies for not being here today. The other big event that occurred this year was the 125th anniversary of the School of Medicine. So our hospital began in 1889, but our School of Medicine began in 1893. Initially, there were not enough funds to begin our School of Medicine. And it was a visionary philanthropist named Mary Elizabeth Garrett who provided $400,000 to begin the School of Medicine. But Mary Elizabeth Garrett believed her philanthropy had to have social change related to it. So she said, I'll give you the money, but there are several changes I want in medical education. Before we began here at Hopkins, medical education was a trade school. You would pay a doctor and you'd work for that doctor, and you didn't really need a lot of qualifications to be it. And Mary Elizabeth Garrett, along with William Welsh, really changed medicine. She said if the med school would only accept people who've already gotten a college degree, they had to know 3 languages, and women had to be accepted on the same criteria as men. That was in 1889. So in that first class of 18 students, there were 3 women. Just to give you a little context, 1893, Harvard Med School started to admit women in 1945. They were always behind us in everything they do. That being said, it really was a wonderful year, and in honor of Mary Elizabeth Garrett, we named our boardroom after her, and just as a small recognition of her leadership and how she changed medicine in this country. Our hospitals are still really highly ranked. Our Johns Hopkins Hospital is ranked number 3. They continue to make errors. We're hoping this year that we're able to move up at least 1 position, if not 2. Our Children's Hospital is ranked 8th. Our School of Medicine is ranked 2nd. If you add that all together, we're clearly the top academic medical center in the country. We're very proud of the care we provide on a daily basis, and for the great people. It's all because of the nurses and the physicians and staff that work at our hospitals. We're very, very thankful for the care they provide to our patients. But today we're going to talk about science, and one of the things that I am a physician scientist, and one of the things as I think through how we cure diseases have always struck me was that if you don't understand basic biology, you cannot understand why diseases originate, and therefore you can't actually find the cures of those diseases. And so I, early on in my own career, dedicated myself to understanding how cells work, how proteins work in the cell, what causes the disease, what's the normal physiology of a cell. And at Hopkins, those 20 Nobel laureates, many of them worked in very basic science, because that is where you make the true breakthroughs that lead to cures of disease. And we decided that we were really going to invest in our basic scientists. We were very lucky to recruit, and you'll hear from him today, James Berger from the University of California at Berkeley to come work here. He has become the director of the Institute for Basic Biomedical Sciences. In that group are all our basic scientists, and they're the ones who really think about how things work. We have some of the best in the world doing that. We're going to put a $100 million investment into our basic sciences over the next 5 years. And we're very glad, James, that you were able to join us. And I think when you hear him today, you'll understand why we're going to really do special things in the next several years. And I'll give you one example of what we've done just this past year. Andy Ewald is a basic scientist. He's a cell biologist, very interested in cancer. And for those who don't know, in breast cancer. So for those who don't know, breast cancer normally occurs in the ducts that produce milk. And those are the cells that turn cancerous. Very often they cause breast cancer. Well, it ends up that around those ducts are muscles, and those muscles are what helps a woman produce milk and pump the milk out. So that muscular layer was thought to just be present. If you think what kills people from breast cancer, it is not the breast cancer there, but it's the spread of the breast cancer to other parts of the body, the metastasis of the breast cancer cells. And for a lot of years, it was thought that those muscle cells just sit there and the cancer cells just break through and then go to the rest of the body. What Andy showed, and this is a nice picture, is that the muscle cells are actively trying to prevent that. And when a breast cancer cell tries to break through the muscle cell reaches out and grabs it back and brings it back. And that's a picture of it right here. That red cell is a cancer cell. The green cell is a muscle cell reaching out and drawing that cancer cell back, trying to keep it from metastasizing. If you understand that, you understand, well, maybe one of the reasons they metastasize is that the muscle cell is not doing what it needs to do. And so Andy has a new whole line of research trying to understand why in metastatic breast cancer, those cancer cells are able to escape the muscle cells, keeping them in the ducts. Another great thing that we're looking forward to this year is proton beam therapy. So radiation therapy has been around a long time. It's really important to treat many, many types of cancer. One of the limitations in the effectiveness of radiation therapy is that the photons, or radiation, not only kill the cancer cells, but they kill and damage the cells around the cancer and all the tissues on the beam path, right? And the toxicity of that is what limits our ability to treat many types of cancer. Proton beam is a type of radiation therapy that gives much fewer amount of damage around the beam and actually directs the protons straight to the cancer. So it's really important in types of cancer where you don't want to damage anything on the path, such as cancers in the brain, childhood cancers, cancers around vital organs like lungs and liver. So it's been very effective in many types of cancers. We are opening our proton center at Sibley Hospital in Washington. It's going to be a magnificent facility. It'll open in this October. It'll treat brain cancers of many types, of pediatric cancers, and newly evolving roles in many different types of cancers. As I said, we're looking forward to treating cancer in Washington, D.C., and around the world. And this will be a place where people really come from around the world to get their cancers treated. We also had another great gift this year. So the leadership of UAE provided us with a $50 million gift to build a stroke institute. The institute is dedicated to better diagnosis of stroke, better treatment for stroke, and better rehabilitation for people who've had stroke. We're using things like artificial intelligence to teach the MRI machines to more easily detect stroke at diagnosis. We also are building a center in Abu Dhabi to help people in Abu Dhabi get the same care that they can get here in the States. Another first this year was from our plastic surgery team. These plastic surgeons are dedicated to doing transplants that other people can't do or haven't done. So Andy Lee and his team thought about what we can do to help our wounded soldiers. For those who you don't know, in Iraq and Afghanistan, as one of the biggest damages that hadn't occurred in prior wars were damages of the penis and scrotum in our soldiers. That's because as the Humvees drove around and these IEDs were blown up, they blew up right underneath the Humvees. And if you think about it, they ruined the perineum of soldiers. And many soldiers lost their penis and scrotum, that whole part of them. And it's a real debilitating deficiency for these people. So Andy's team practiced 5 years and did the first total penile scrotal transplant in a wounded warrior from Afghanistan this past year. And the patient's doing really well and regaining function. So we want to thank Andy and his team. It's really amazing. 9 surgeons, 14 hours to do that surgery, 5 years of practice. So it was an amazing event. Finally, the last thing that we did, I think that was I wanted to cross this year, was a new type of total ankle replacement. Jim Fricke and his team did in orthopedic surgery. We were one of two that put in this new ankle replacement. And the neat thing about this ankle replacement is because of the way it's used, you don't have to cut away a lot of the prior bones. So, and one of the problems with ankle transplants, they last 15 years, 10 years. And so if you get them young, in a couple years, you need another one. And the old ankle replacements, you had to chop away so much bone, it was really hard to do another replacement. This new replacement, you chop away very little bone. And so if someone needs another replacement in 15 or 20 years, they're able to get one. And so it's a really neat thing that Jim Fricke and his team did this past year. So as I said, Hopkins is a place of innovation, of people who are dedicated to changing the care of our patients, to advancing science in the hope of cures of disease. And that's what we're going to hear about today. First, we're going to have James Berger come up and tell us a little bit about the basic research that goes on at Hopkins, and then we're going to have a panel talk about how we help discover new therapies and drugs for patients. So James, please come up.
Okay, thank you very much. Can you hear me in the back? Okay. Is that all good? No, you can't hear me? Sorry, it's good? Okay. You're playing, are you playing? I can't tell you playing. Okay. So, so at any rate, thank you very much for the opportunity, Paul and Ron, to be here today and tell you a little bit about basic science and how basic science contributes to the general health mission at Johns Hopkins and elsewhere. And in particular, I'm going to talk about how basic science is concerned with the drug discovery process. And so what you're looking at on this screen is basically a protein from one of your cells bound to a little piece of DNA. And as this swings around, if you squint really hard, you might see a little fleck of red buried in the middle. Just went right past that, right? So, and that's actually an anti-cancer drug bound to this particular enzyme DNA complex. Now, that may not make a whole lot of sense right off the bat, so at the risk of causing you just a little bit of pain, I'm going to take you back to school for just a minute and sort of give you a grounding on how molecular biology has sort of changed the way we think about the molecules in our body. And as shown here, as we know, the fundamental building blocks of all matter, of our cells, of solid matter in the universe, are atoms, right? Atoms and elements. And so that includes things like carbon, oxygen, hydrogen, etc. And when you take those atoms and put them together with various kinds of bonding rearrangements, you can make molecules. And molecule is just kind of a word that describes a collection of atoms that are grouped together in some way. And molecules can be small, they can have a small number of atoms, or they can be quite large, have very large numbers of atoms. And a molecule that has a lot of atoms, and that you've all heard about, is DNA. So DNA is what we call a macromolecule, macro for big. And DNA, you have about, on the order of about 60 to 100 billion atoms worth of DNA inside every one of your cells, right? So quite a lot of atoms strung together in that molecule. Now, DNA is what makes you, you. It contains all the genetic information that distinguishes you from others. It distinguishes us from dogs or cats. And the way DNA does this is it has instructions encoded within it that basically make another complicated set of molecules called proteins. And proteins are constructed of small molecules known as amino acids strung together in a long linear chain, and then those chains fold together like a sort of three-dimensional jigsaw puzzle to give you a particular protein or a particular enzyme that has a unique shape or a unique function. And those proteins are basically what do everything that makes your cell work the way it does. They communicate with other cells, they harvest energy, they replicate cells, etc. And it's the combination of the DNA in you and the proteins and then some other molecules I won't go into that ultimately give rise to the cells and that make you, you. And so with that, I can now sort of go back to this little movie, and you'll see a zoom in here. This was that molecule I showed you earlier, bound to DNA and anti-cancer target, and there's the drug sitting right there in the middle. And so what we have today are tools and technologies that allow us to see these molecules literally down to the level of individual atoms, and we can tell exactly where each atom in a protein is, each atom in a DNA is, and then how each atom of a drug might interface with those kinds of molecules. And so that seeing gives you a lot of very powerful information to basically improve drugs and to help make drugs better. So to sketch out a little bit of the drug discovery process, I'll illustrate this with the pieces of a funnel here. So there's many aspects to drug discovery in which basic science plays a role. The first is that we have to, when you have a human health disorder of some kind, you have to find out what its roots are. What's causing that disease? Is it a pathway that's broken? Are there proteins that are broken? And this is sort of where the disciplines of genetics and cell biology and scientific fields of inquiry such as those come into play. Once you know what's causing the basis of a disease, you then have to find something within it that you think you can tickle or trick or toggle or play with to basically change that disease state back into a normal physiologic state. And this is where we have to find targets that we can hit with small molecules. These are usually enzymes again and proteins. And this is where things like biochemistry and again elements like genetics come into play. Once we think we have a good target, then starts the process of actually trying to create a drug that will do what we want to do. And there's a lot of different scientific disciplines that go into this process: chemistry, pharmacology, structural biology, just to name a few. And these many iterative cycles of sifting through different kinds of molecules, trying to find something that might specifically target that protein that we're interested in. But once we get something that we think is going to work, then we can actually start to do clinical trials with that. And that involves things like patient selection. And if all of that goes as planned, at the end of the day, there's a little tiny spigot at this funnel and you get a drug out, right? So this huge foundation of basic science, which funnels down into each drug that we might have to take during the course of our lives. Now in this process, historically, academia really played a predominant role in helping with this early stage of finding disease roots, right? Sort of figuring out what's going wrong and why it's going wrong. And all the other elements of this funnel were largely conducted by the pharmaceutical industry, right? But now, thanks to both technological advances that have come into the academia world, as well as marketplace pressures on the pharma sector, it's now becoming increasingly common for academia to actually play a fundamental role in many of these early stage efforts of drug discovery. And I think what's really exciting about the times we live in is that the tools are only getting better for us to be able to do this. All right, so what do basic scientists actually do? And so there's many different features to this. One is that although we don't treat patients, no one would trust me to treat a patient, and that's perfectly okay. We do create the cures that ultimately physicians can give to their patients, and it's the groundwork that we lay that does that. The other thing I think that's important for the general public to know is that we work for you. We are taxpayer-funded biomedical researchers. We receive the vast majority of our funding from the federal government, and it's your tax dollars that really drive those cures. And to illustrate that, I'm just going to show a little bit of history of the National Institutes of Health, which is the premier funding agency that gives us a lot of our resources. So if you take the cumulative amount of money that the federal government had given to the NIH from its inception around late 1930s to say the mid-1970s, that totals to about $21 billion. But out of that research has come innumerable cures through the pharmaceutical sector, and also through an exciting new sector we call biotechnology. And that biotechnology sector alone today is approaching a valuation of close to $1 trillion in terms of the products it makes, the people it employs, the science it creates, et cetera. So in that regard, those investments that your grandparents might have made or that you made at some point, those have had an enormous impact on the health system. And I want to point out that Johns Hopkins has been leading this charge from the get-go. In fact, a lot of the technology that today's biotech sectors are founded upon, namely the ability to cut up little pieces of DNA and stitch them together and make new antibodies or new genes, things of that sort, rests on discoveries that were made at Johns Hopkins back in those 1970s, for which people like Dan Nathans won a Nobel Prize. And that tradition of excellence is still ongoing today, whether it's in cancer immunotherapy or repurposing existing drugs for new diseases. Now, along these lines, we still need help from the public in a number of different ways. Today's annual budget of the NIH is roughly about $37 billion, which sounds like a lot of money, and it is. And that money funds about 50%, little less than that, of the basic science that goes on in the labs of doing the type of work I've been discussing. But there are some problems with this, and one problem in particular is that funding level has been flat. In fact, it's been worse than flat. It's actually dipped, and it's only just now starting to climb back. It's been flat since about 2003, and that's really hampered our ability in many instances to sort of move forward. Another problem that we face as basic scientists, is that a substantial amount of money, the portion of money that goes to the basic sciences has been decreasing over time. And then finally, I don't think it's a secret to say that what the NIH funds, they're fairly risk averse in what they decide they want to fund. That makes sense. They don't want to waste taxpayers' dollars, so they don't want to fund something that they aren't pretty sure is going to work. But that also hampers our ability to do high-risk, high-reward science. And that sort of collection of issues, if you were, poses a bit of a problem for us. Because if we want to translate our discoveries into cures, well, we can't translate something if we don't actually know how it works to begin with. And so this is where basic science really has its work cut out for it. Now, another component, I will say, of basic science that really derives its support from the public is philanthropy. And philanthropy contributes about 10%, give or take, to the overall basic science mission in the United States. That might seem like a modest number, but that money is actually quite important because it allowed most of the support that comes from philanthropic dollars allow us to take on high-risk, high-reward ventures. And so big breakthroughs can come from those efforts. So now, why support basic sciences at Johns Hopkins? Well, there's many reasons. One is that, as Paul already alluded to, we are leaders in basic science and we are leaders in clinical science. And more importantly, we're leaders in connecting those two together. And it's that culture of that connection between clinical and basic that really is what makes Johns Hopkins so distinctive. As I mentioned earlier at the outset, this is a tremendous time in terms of technology for the basic sciences. The tools that we have in my lab today, I didn't even imagine when I was a graduate student, and when I think what we might have in another 10 or 20 years, it's pretty eye-popping. So it's really a great time to be doing science. And I just would like to close by saying that as basic scientists, we are really grateful for all of your support at so many levels, whether it's support to the NIH, support to Johns Hopkins directly. And so we really look forward to working with you in the future on this front. So I'll stop there and introduce our next speaker, who is Dr. Bill Nelson, who's the director of the Cancer Center. He's going to be the speaker. Thanks.
Well, good afternoon, and thank you all for joining us this afternoon. We feel very welcome here in Palm Beach and have been every year. I've been in cancer medicine for 30 years, and it's remarkable for me to sit here or stand up here and think that 30 years ago, we didn't have someone like James Berger helping us make anti-cancer drugs. What an error that was historically. What we were doing instead is we had embarked on this strategy across the country of screening for cancer drugs. Screening drugs, chemical compounds from nature, actually found some good ones there. Some from tree bark, a famous drug, some from a sewer in Milan, you can't make this up, another great drug, and some from the chemical industry, some from the photographic industry. We screened them with the same logic in mind. We tried to see which of these whole slew of chemical compounds could we put on a cancer cell growing on a dish in a laboratory that would kill that cancer cell dead. And they all work pretty much the same. The more of these drugs that we put on these cancer cells on the dish, the more of them we could kill. The clinical exercise was then, how much of this could we give a person? Could we give a person enough to kill the cancer cells in their body without doing too much damage? And if you think about the ones that sort of worked, they still do a lot of damage. They lower blood counts, they drop people's hair on the floor. They're very hard drugs to give, much harder drugs to receive, I can tell you. And this was a tough era, and it wasn't very efficient. Drugs that entered clinical trials had a 9% or less chance of ultimately proving beneficial and being approved by the FDA. And the people who participated in these clinical trials, remember these are people with bad cancers that are growing and no standard treatment options, they had a less than 5% chance of any benefit at all. A very heroic collection of people. But what James Berger has talked about has fueled an explosion in anti-cancer drug development. And Johns Hopkins is the perfect university to think about this. There is a breadth of expertise across disciplines, critical minds, not traditionally involved in the cancer problem, but we can now bring together in teams to build new anti-cancer drugs more efficiently, more rapidly. 25 years ago, we were approving one anti-cancer drug a year from the FDA. For the last decade, it's been 10 per year. Cancer outcomes are getting steadily better and better and better. Something on the order of 2.5 million lives have been saved over the last 25 years by advances in cancer treatment, and it's because we're bringing people together like James Berger. Now, cancer clinical trials seems very ethereal. We're doing clinical trials. It seems like a science experiment of some kind. But for the people who participate in these types of clinical trials, this is their treatment. So what we thought we'd do today is introduce you to someone who's had cancer, who's participated in these kind of clinical trials, Mr. David Gobin. Mr. David Gobin has served our country and served our community in Baltimore. He was a Baltimore City police officer for 28 years, headed up the K-9 division. If we have a little time, we can get into a discussion about Shepherds versus Malamutes as the best K-9 dogs. He can wax poetic on that, actually. But he discovered a few years ago that he had lung cancer. And believe me, he went through all the treatments that we had in our toolbox: surgery, radiation therapy, chemotherapy, and the like, but his cancer returned. He ultimately participated in a clinical trial, and with that, I'm not going to tell you any more of his story. Mr. Gobin, why don't you come up and join us and we can have a chat. So why don't we start at the start of your cancer journey. You are a brave and tough guy. To protect the citizenry of Baltimore, you put yourself in harm's way routinely over most of your life. What happened when you heard those 3 words, "you have cancer"? How were you feeling? What did you hear? What was your response?
My initial thoughts, which I can't repeat, it wasn't real nice, but I think a lot. I said, cancer, you got no idea who you picked a fight with, because I'm going to win this fight. I know I am. But I figured I need a partner in this fight. And I said, Johns Hopkins is probably the one I want to bid, and thank you very much. We eventually beat that fight.
So you were ready to fight back, and you had, and I'm sure people talked to you about what the treatments that were involved and stuff, but did anything really prepare you for surgery, radiation therapy, chemotherapy? That's not just fighting back, that's really fighting back.
Prepared for it? No, but when you don't have a choice, you do it. I mean, I wasn't afraid of it. Let's just do it.
What were the toughest parts of that treatment?
The toughest part was I would have to say the first clinical trial. That drug was tough. It was real tough, but I'm tougher. You know? I don't want to get in line and volunteer for it again. But when that one didn't work, my tumors grew, and I said, all right, this is all between a whole lot of other stuff. They had another phase 2 clinical trial, chemotherapy, which wasn't as bad as the first one. That didn't work either, and tumors grew, and that was it. And they basically told me, that's it for the chemo.
So let's back up a second. excuse me, and when you were presented the option of participating in a clinical trial, and I think most people, until they've been in your shoes, facing the choices and the experiences you've had, think of clinical trials like a biology experiment. They're dissecting a frog in high school or something. How was it presented to you? How did you think about participating in a clinical trial?
Well, I always said I'll do whatever they want me to do, and I'll take whatever they give me. But I kind of thought with the immunotherapy, I said, this sounds like an experiment. I'm not the smartest guy in the world, but it sounds like an experiment. And it sounds like it's something right up my alley. And I'm excited to do it. But the thing is, I always thought that if it doesn't work for me, maybe they'll find something in me that'll work for somebody else. And that'll definitely make it worthwhile.
So that's one thing about providing cancer care that's always very inspirational about people who participate in clinical trials. As you heard what he just said, there was this sense that whether or not this clinical trial provided a direct benefit to him, the people we find participating in clinical trials are very conscious that the knowledge gain might benefit everybody. Somebody coming later, somebody who you don't even know might benefit from your experience in a clinical trial. The other thing we hear from people, I've heard from him several times, which is, we're heading to the 50th anniversary of Neil Armstrong walking on the moon. Many of our people who participate in clinical trials have a good sense that they're out there in front as an explorer. Did you have those kind of feelings?
Yeah, I did, because that first immunotherapy, I was the 96th person ever to get that drug. It had no name. It was MDX 1106, which now you might know as...
Sounds like a mission, doesn't
Yeah,
it?
but
MDX 1106.
but now it's Opdivo. I didn't know that at the time it was ever going to get a name. I said, yeah, I was told it was for dosage only. That's what they were doing on me. They
Trying
wanted
to find the right dose to get.
right dose. But you know what? They don't know if it could do anything, but hey, I'm a fighter. Let's fight. I might be the first one. Who knows? You always got to have hope. Can't quit.
So as you were being treated on this mission with the MDX compound, MDX came from Medirex, which ultimately was bought later. But as you were participating in this was your treatment. This wasn't an experiment. How did they follow you? How did they manage you, if you will? Were they using scans? Did they work with you like this was your treatment, or did they work with you like this is an experiment and they don't care about you?
Oh, God, no. I've never felt that Johns Hopkins didn't care about me. That's impossible. That would never happen. I know it wouldn't. In my head, I know it wouldn't. And, yeah, they followed me closely, very closely. I think they documented everything. They wrote everything down. I had infusions every 2 weeks, and my gut compared to chemo, they were nothing. They were a walk in the park. And I had CAT scans every 2 months.
Yeah, it turns out the side effects of this MDX compound were substantially different, wouldn't you say, than the
They were
chemotherapy?
basically none. I still suffer from side effects from the chemotherapy that I have not had any chemotherapy in over 8 years. And I still have side effects from that chemo. And I have nothing new from the immunotherapy, and I think that's wonderful.
Well, one of the things another thing that we've talked about over a while is that this was a very new kind of treatment, and we weren't even sure, I think, how to follow whether the treatment was really working or not. Very oftentimes in cancer medicine, somebody has cancer that we can see on a scan, and the logic was pretty straightforward. If it was shrinking, then that was good. If it was growing, that was bad. But we started to wonder with this new kind of immunotherapy, what would happen if all kinds of immune cells went into the cancer and tried to destroy it? Would it look bigger, or what would it look like? So what happened with you?
The first scan showed growth. And when growth occurs, you're taken off a trial. Right? But this one was so new that Dr. Bramer said, let's see. We'll see what happens in the next scan. We're not going to take you off because of growth. We don't know. We just don't know. Well, my next scan, which was, I can't remember if it was a month or 2 months later, I get a phone call and they said, you got significant shrinkage. I said, whoa. All I ever hear is, your cancer's growing, your cancer's growing. Now they call me up and say I have significant shrinkage. And they said, did you hear what I said? I said, yeah, I have significant shrinkage. I have no idea what that means, but I'm yard sailing. I'll get back to you later. You know, and
Did Julie Bramer sound excited, though?
oh my God, was she ever? But, you know, I very quickly realized that, you know, maybe there's something going on when NBC Nightly News shows up doing an article on me, and Stand Up to Cancer wants to do an article on me, and they're doing a convention in Chicago, they're talking about me, and I said,
He's a celebrity now.
me, you know what I didn't do anything but sit there. I swear to God, I did nothing but sit there and let them put it in me. And that's all I had to do. You guys did the work.
So how long ago was that?
Okay, now, remember, this is stage 4, non-small cell lung cancer, which has a very short lifespan. That was, I had a 2-year trial. I've had no treatments now for over 6 years. And if I get this right, I get the PD-1s attached to my T cells, they're still working. They don't know why, but they're trying to find out.
So we, you know, in cancer medicine, we don't use the word cure very often, as he'll tell you. We certainly, for lung cancer, use the word incurable quite often.
Oh, yeah.
And yet you get out 6 years without any breath of the lung cancer, and you can start occasionally dropping the cure word pretty often, I would think, and I hope so. So tell me, if you had to participate in a clinical trial tomorrow, would you?
Yeah. Yeah. In a heartbeat.
And what would your advice to other people who've heard those 3 words, you have cancer, and somebody's mentioned the notion of participating in a clinical trial, what would you tell them?
Don't hesitate. Get in line. Start tomorrow. You can't wait. You can't wait. You can't be afraid. You can't quit. You can't give up. You got to fight. Fight. And I've had countless people call me up when they hear my story. And I tell them one phone call. Kimmel Cancer Center, Johns Hopkins Hospital. Call them. If you have lung cancer, I got the oncologist for you. And, you know, I tell them research is wonderful. Look what it's done for me. That's research. Go to Hopkins, they're doing the research.
And so what have you been up to in the last few months? What have you been doing with your life now?
Well, I'm not shoveling snow.
There's something about Florida, isn't there?
Yeah. We'll go back to Maryland to spend the summer up there. But I'm not going back until it gets up to at least 60. And when it gets below 60, we'll be back down here. And we find out it's 40 degrees here.
And then my last question for you, shepherds or Malamutes?
Shepherds.
So, why?
No questions about it. For my buck, I've never had a Malinois, but I've seen them. They had them towards the end of my career.
He told me it was political, right?
Political.
Imagine that, yeah.
And they just didn't have, I think they had the courage. Like I said, I'm prejudiced. They didn't have the courage a shepherd had. You know, and
Well, thank you.
the loyalty, my dog would have died for me in a heartbeat.
Well, thank you for the courage that you have.
Oh, my God.
You're an inspiration
It was
to
my
all of
pleasure.
us and all of us here, and thank you again
No,
for
thank
joining
you.
us.
Thank you.
And now we'd like to assemble a panel to talk about the new smarter ways we develop drugs that has more people like David Gobin benefiting in clinical trials and more drugs being delivered to clinical care. First, James Berger, who you already met, Mariki Leho from our Department of Radiation Oncology and Molecular Radiation Sciences, and Srinivasan Yegnasubramanian from our Department of Oncology. I'd start by hinting that pretty much everyone you see here is very different. In fact, it's striking how different your backgrounds are, how different the perspectives that you bring to anti-cancer drug discovery are. How did you get into biomedicine and cancer medicine, Vasan? Where'd you come from?
Well, I guess for me, the journey started when I was in college. I was at Duke University mainly because I liked the Blue Devils as a college basketball team. But while I was there, I found my love for science and technology and was trying to think about how I can develop that, and ended up in the School of Engineering thinking about how we can bring science and technology to bear on solving problems. And as part of the coursework there, I had the opportunity to take project classes that took me to the Duke University Hospital and talk with teams about what kinds of things they needed, and we were able to come back to the labs and classrooms and try to think about how to build those devices to fill those needs. And once I saw that world of medicine and biomedicine, I was hooked. That's how I got into medicine. And eventually that brought me to Johns Hopkins for medical school. And in medical school, as I confronted cancer as a problem and saw the clinical care of cancer patients, I think I really found my calling, and thought I need to spend the rest of my life trying to think about how to find better medicines for that disease. And I guess that's how I got into it.
Well, Mariki, an engineer from Duke, not exactly your experience, came from Finland. How did you get here?
Yeah, so I was a medical student in Helsinki, reading textbook after textbook after textbook. And you think that by all that knowledge, we'd know how we can treat all the diseases, but that's not the case. And especially, I realized that when I started to ask questions, what causes these diseases? And then you understand suddenly that we do not actually know all of it, and we need to learn more in order to be able to better treat. So for me, that meant that I started a summer research project in the lab, working on a cancer biology program, and that is what drives the growth of the cells. And I've never left the track. I've worked on the same problem basically ever since.
So then did you get rid of all those textbooks, or?
Yes, I hated them.
So James, we heard a little bit about you. You're a structural biologist. How did you come to the biomedical research, cancer medicine?
So I was on the faculty at Berkeley for about 15 years as a very fundamental basic scientist. But when the opportunity came up to move to Hopkins and start to think about new ways to take some of the discoveries we had made at a very basic level and partner with people who actually thought about practical applications and doing something more with them, that was just too good to pass up.
So Mariki, President Daniels actually mentioned this briefly in his remarks. You've had the opportunity to take an idea from a discovery idea, finding a chemical entity, a small molecule, a chemical compound, that did something biologically that you were studying, and then to begin the process of making an anti-cancer drug out of that knowledge. And you've gone, 2/3 of the way through the process. You've had to cobble together diverse expertise, diverse funding mechanisms. How has that journey gone? Tell us a little bit about how you've done that.
I'll start from the beginning. So in the beginning, the goal was, okay, can we activate a major protein that's really blocking cancer cell growth? And how can we do that chemically, meaning using these small molecules, putative drugs? So we searched after thousands and thousands of molecules to try to identify one. And we did, and it worked. And it was truly effective in killing cancer cells. But then our research ran into a stumbling block because we realized that actually the molecule is not working exactly as we thought. It didn't hit the anticipated target, or it did, but we realized there's something else beyond that. There's another pathway that is really important to block in the cancer cell's ability to grow. And that was really the crucial moment. So it was obviously, it's a fantastic moment when you discover something new.
This wasn't just a drug that just killed a cancer cell. It did a very specific thing that you figured out.
Exactly. So that required a lot of painstaking basic science work. And we were lucky because it was successful. But at that point of time, you are at the watershed. What are you going to do that? So science-wise, it was really cool and nice. But then the next question is, can we push this forward? And what would be the means and tools how we can accomplish that? And it wasn't straightforward at all, because my background was in the basic science, in cancer biology, as in the medicine, but not in the drug discovery pathway itself. And that meant, okay, now we need to reach out to the experts in this field and collect new kinds of teams, including.
Yeah, so it's interesting. Historically, right, you would have published a paper, which you did, communicate to other scientists what you've learned, and you would have hoped that somebody in a drug company would have read the paper and said, this is a good idea, and make a drug. As James Berger hinted, the drug companies aren't doing so much of that anymore, and so you needed to work to push it even further by bringing together this expertise. How much of it was available at a university like Johns Hopkins?
Well, a university like Hopkins is a fantastic environment exactly for this purpose, but you need to find out whom you need to reach to. So we needed to engage with medicinal chemists, with pharmacology people, with pharmacodynamics people, toxicology people. And we were out here. If you can bring me back, thank you. So it was a lot of kind of just networking, asking questions after questions. What do we need to do next? What do we need to accomplish? There's actually one very important aspect right here, which is that, from academia, we don't have the resources to make a truck like a pharma company does. That's hugely expensive. The ticket is probably around at least $10 million when you are successful, when everything works. So we can't do that. But what we can do is that if we have the relevant intellectual property, meaning patent, around our small molecule, we can then engage additional partnerships and reach out. And that was then the kind of a next step and hurdle that we had. Oh, okay, where are we going to go? Who are we going to ask for help? And I asked help a lot, meaning approaching small pharma, large pharma, biotech company, business angels, venture capital companies. And then finally, something came true. And it actually came through a Hopkins partnership with a major life science venture capital company. And they had just come into Hopkins and asked and told Hopkins, we want to engage into the basic science discoveries. We want to be able to translate these discoveries into the healthcare, into the clinical uses. So we were really fortunate of being able to receive funding from them, and that's the piece with which we are.
The attractive notion here is that people like Mariki in an academic center using the resource of the university are building more and more value, which make us more and more attractive to these investment opportunities. So Vasan, if you think about cancer almost with an engineering perspective, engineers like a problem with specs on it. We've known forever that cancers are to sort of acquire defects in genes and gene function. It took us forever to figure that out. We knew that for a few genes, but at this point we can measure every gene in every person. What is that technology doing? What is that teaching us, and how is that going to change anti-cancer drug discovery?
Yeah, you put it in a good way. To try to break that down into an engineering problem, it's helpful to figure out what the size of the problem is. If you think about the genetic information in the cell that James talked about, this DNA molecule, that DNA molecule is a really long molecule, and it's made up of A's, G's, C's, and T's, like you would have heard about in high school biology or in like science fiction movies like Gattaca. But the way that carries information is that they're all sort of strung together in different orders, and the order in which that they occur gives the cell information about what instructions that it needs to follow. And the size of that instruction manual is actually quite huge. So in our human cells, each copy of our DNA has 3 billion, essentially, of those letters strung together like that. And there's 2 copies in most of our cells. And if you think about that, what that means, for one of the textbooks that Mariki was talking about, each chapter might have something like 30,000 letters. And so we're talking about 3 billion divided by 30,000 would be something like 100,000 of those chapters. So that means something like 5,000 textbooks. So we had a tough time reading a few textbooks in medical school. Our cell is constantly reading this instruction manual and doing the instructions from it all the time, except it's this huge magnitude of information. Now, what's very interesting is that these cells also have to figure out how to do something else, which we never had to do, which is that they constantly have to copy that instruction manual when one cell becomes 2 cells and 2 cells become 4 cells. They constantly have to copy and copy. All those 5,000 textbooks worth of information have to constantly be copied. And it has to do so in a way that there's very few mistakes. And what we find out about cancer is that what's happening is that occasionally the cells will make mistakes in copying that DNA at just the right places. And those mistakes that happen can give the cells new properties that allow it to break the rules that normal cells usually have to live by. And that's one of the ways that cancers can form. Now, what we can try to do now, using technologies that harness innovations in many different fields, engineering, molecular biology, chemistry, computer science, data science, and put these innovations together in new technologies that allow us to sequence or read all of those letters that happen in a given cell's genetic code. And we can do that from, let's say, a cancer specimen from a person and also the normal cells from that person, and compare them and pinpoint exactly which of those letters in that 3 billion long letter stretch have become different, have gotten mutated. And by understanding those and doing that across many people, thousands and thousands of people, we can try to figure out what patterns there are and what kinds of changes in that instruction manual occurred in any given person's cancer or group of people with the same types of patterns. And that information can be translated to the fundamental observations that we need to figure out how can we kill those cancer cells, but not the normal cells. And that's really how that science can play itself out in useful ways.
You talked about Gattaca, sort of science fiction. This getting the sequence information for all the defective genes, this is something that we do for patients that come to Johns Hopkins. If they get a biopsy, if they get their tumor removed, this kind of sequence analysis, this does seem like science fiction, we do for the majority of them, because it may be that one person's cancer needs one particular treatment, another person with what seems to be the same kind of cancer might need a different kind of treatment. That information is in the sequence that we use for clinical decision making. It's really astonishing. Now, these differences in genes, these create defective proteins. You took us through a tutorial. The exam is later. Before they let you have any food, you have to pass the exam quiz. But there used to be some of these genes we would discover would be clearly a defective gene driving cancer, and then everyone would throw up their arms and say, conventional wisdom, you can't make a drug against this target. You've spent your life looking at these proteins. You showed a picture, of course, of one of the empirically discovered anti-cancer drugs. We did not know how this drug worked for 30 years, and you helped us learn how it worked. But is there an undruggable target? Can you tuck one of those little red things into any part of a protein? How's that work now?
So I think, when you say undruggable, I won't say that there's no such thing as I'm sure there are some things which may not be druggable, but because of the tools and technology that we now have, the repertoire of what we can think about drugging, as it were, is much greater than we ever would have thought. What we now know is that those pictures that I showed, they're pretty. We can look, and again, we can see where every atom is, and in a 100,000 atom complex, we can tell where each one is, what each one is, and so we have all this immense information. But it turns out proteins don't just stand still while we take their pictures. We have to actually force them to stand still so we can take a picture of them. And when they're in your cells, they're constantly moving around. They're constantly jittering and opening and closing and doing things, moving about. And when they do so, they actually expose new features, cavities, crevices, things like that, that now you can imagine dropping a small molecule into them. And as we know more about what these enzymes and protein complexes look like and how they move in real time, we're getting better at designing specifically experiments, assays, if you will, that we can use to find small molecules to hit them and turn them on or turn them off in very specific ways. So I think that the repertoire of what we can now tackle is just much more exciting than we would have thought.
And of course, this is something that the pharmaceutical industry is not able to do. You need the firepower of discovery researchers at academic centers solving these fundamental problems. That's why playing a disproportionate role in building anti-cancer drugs now and I believe for the future. Tell us about the next wave of technologies. I mean, you would get a protein, a small piece of one perhaps, like you say, you'd force it to hold still so you could look at every atom. What are you doing now?
Right, so the challenge that the pharmaceutical industry faces, which is the same as academia faces, is that we have so many different proteins and so many pathways, and we now know that so many of these factors we're interested in actually don't just do one thing. Sometimes they do 3 things or 5 things. And so the number of, you can't, no one company can even begin to place a bet on every possible thing a cell does. So what you have in academia is you have the scale of lots of people taking lots of bets, lots of shots on goal, and I think that's one of the powers of what academia can do. The technologies that are now available, that picture that I showed earlier, that particular structure of that enzyme DNA drug complex, we obtained that using a method it's a technical term. It's called X-ray crystallography, and it's kind of new agey. But
use that with your grandkids.
That's right. It's kind of new agey, but you actually have to grow crystals of the protein you're interested in, and then you shine X-rays on it, and then from the way the X-rays interact with the crystals, you can deduce the structure. It's very laborious, very time-consuming. It took us over a decade to get that structure. With the new technologies that are coming about with electron microscopy, we could solve that same structure in weeks or months. And moreover, we don't have to grow crystals anymore, that's the big bottleneck. And so tools like that, there's equivalent tools in mass spectrometry, all different kinds of imaging modalities or techniques. And then, there's tools in the kinds of experiments that, say, Vasan uses in terms of sequencing, which you just alluded to, where we can get so much more information in such a shorter amount of time from so much less material. That's what makes it really exciting time.
So with this, the sequencing technologies have also grown to the point where we're not just looking at the A, T, G, and C, because of course, not every cell uses every gene every minute of its life. Eyeball cells use different genes than fingernail cells, and cancer corrupted in that space too. This is epigenetics above the gene field, and you're the world's expert in it. How does that work? How's the technology going to get us that? How bad of a problem is that in cancer?
Yeah. So maybe I can back up and frame that a little bit also. So, we talked about the lots of information in these DNA molecules in our cells. The other really strange thing about it that you have to think about is that the DNA is so long that those 3 billion base pairs, if you just think about the physical size of them, in one cell in our body, if you stretch that out end to end, it's actually about 6 feet long, okay? And what's amazing is that gets folded and crumpled and put into a little cell that's really tiny, and that's really hard to think about. So if you think about it in one way, if the cell was the size of the dot on an eye on a printed page, that DNA stretched out would be the length of like a football field, okay? So that's all getting folded up and put into that little cell. And the cell is constantly trying to figure out how to read which parts of it to do the stuff it needs to do. Now, what's interesting is that the cell doesn't just fold it and crumple it up willy-nilly and stick it in there. It actually folds it and organizes it in the right ways so that it can access information in the right ways. And that level of coding is really important. So it's the type of coding that we call beyond the genetic sequence, epigenetics, that the cell uses. And scientists all over the world, including in our lab, we're trying to think about exactly how cells operate and organize that DNA to do that. And it turns out one of the ways that it does that is a way that we might organize our own notes. It actually puts chemical marks on different parts of that DNA, or the proteins that bind the DNA, that are kind of like bookmarks or sticky notes on the sides of pages in the textbooks that we would read. And that kind of a marking structure can help it figure out which parts of the instruction manual to read at any given time. Now, normal cells have a rigid set of rules that they follow, so that our liver cells will make one set of sticky notes, our brain cells might make a different set of sticky notes, our eyeball cells might make a different one, just like Bill was talking about. But what happens in cancer is that this level of coding also goes completely awry, and they find out ways to change those epigenetic encoding, or these sticky notes and bookmarks, in a way that they can break a lot of the rules and work only to their own advantage and growth really at the expense of the rest of the body. And that's partly what cancer does. So one of the things that we can do now is to use the same types of technologies that we can use to map the genetic sequence. We can harness them with different molecular biology methods so that we can also see these epigenetic coding. And when we do that, we can compare again what happened in the cancer cells and what happened in those normal cells, compare them, figure out those differences, and then go back to the laboratory and figure out how those differences were made and what things we can target with drugs. And so a whole class of drugs in cancer medicine are coming in this field of epigenetics to try to figure out how we can really manipulate that back in a cancer cell so that it doesn't have those advantages anymore, and we can really kill those cells.
So we really are at the precipice of a new wave of diagnostic tests, new drugs, et cetera. Turn to Mariki. Talk to me a little bit about the way science, like you've done, you made a discovery, fundamental biology, an opportunity to make a drug, you navigate all the way up to a venture capital investment. Talk about funding. What's the role of the NIH? What's the role of foundation? What's the role of philanthropy?
Yeah, so of course, you know, in basic sciences, our bread and butter is the NIH funding, and we are competent in acquiring that.
You guys are really good at it, let me tell you.
It has challenges, it's not straightforward at all. But now, and foundations are super helpful in the sense that, you know, they want to bring in funding towards a specific types of cancer, breast cancer, prostate cancer, and so forth. And that's providing us opportunities to do exactly what James was talking about, the high-risk research. So with the NIH type of a funding, we do, I would say, a little bit conventional type of a science, sometimes even incremental. We hate that word. We shouldn't be doing that at all. But we need to get the money. So with the foundation type of money, we can go for these high-risk projects, which oftentimes they fail. But that's okay, because with every failure, we are still learning something new, and it's taking us to a new set of questions, and that's really important for the field to be able to move forward. So now at the stage when you actually need money to apply your discovery, in this case as a drug, you have to search completely new types of partnerships. And we are not, as scientists, not really equipped well to do that. So of course we have help by our, for example, technology transfer office. So they are the mediators who help us to learn a new language and talk with the patent lawyers, what do we need to do now, and so forth. But it's a lot of trial and error. Takes a lot of persistence, I can say for sure. So I met with 30 different companies and representatives until we finally were able to tap into a real opportunity. But it's doable, and I think, you know, it's something that we should be encouraged to do, because ultimately all of us, we want to see that these discoveries that we make in basic sciences reach to help the patients and in the clinical uses. That's really the dream that we have.
So this won't surprise any of you joining us today in the audience, but this kind of thinking, this kind of talent is highly coveted throughout our country, both at academic centers and commercial centers and the like. And believe me, there are people somewhere that would pay them more money to join them than we can pay them. Why are you still at Johns Hopkins, James?
It's the culture. It's that, you know, what I had heard about Hopkins before I moved there, before I moved here, which was this unique environment that had the ability to have clinical scientists talking to basic scientists to actually basically be more than just the sum of your parts, right? That's what Hopkins was about, and that's true. And so that's why it's such a great place to be.
Mariki, you're from across the ocean. Why are you still at Johns Hopkins?
The environment is wonderful. What I truly appreciate is the collaboration spirit that exists among Hopkins faculty members and staff and all the teams that we have. And it's really the ability to reach out to the experts and somebody willing to work with you and helping you with the problem that you may have. It's exceptional. The other piece that I'm really, you know, aspired about is the ability to translate, meaning that we have the desire and the goal and the willingness to push forward and to be able to make the translation, because making the translation from a basic science discovery into clinical use is not straightforward at all. It sounds so easy, but it's a lot of hurdles. But at Hopkins, I think there's a lot of precedents and demonstration that we can do that, and we aspire for that, and we try to do that.
And Vasen, of course, as I kid you all the time, the Hopkins lacrosse team's pretty good. The basketball team isn't quite of the caliber of the Duke Blue Devils that you love so much. Why are you still here?
Well, really, it's really the same reason that brought me to Hopkins to begin with. I remember more than 20 years ago now when I was interviewing for Hopkins for medical school. I got the opportunity to meet students and faculty, and student after student, faculty after faculty member that I met. They were impassioned about taking on challenges really that were facing medicine, facing society, and working together to solve them. And I was so excited and energized by that, and I got the good fortune to come here. And since then, that same feeling has been there throughout my entire 20 years at Hopkins now. And it's just been fantastic to work with the brightest minds, James, Mariki, Bill, others, in trying to think about how we can work together to tackle the big problems in medicine.
So I think that what you've heard is if you can, in the setting of a great university, bring the best minds around a noble purpose with partners like inspirational folks like David Gobin, I don't think there's anything we can't ultimately accomplish. And I think we will ultimately cure cancer, and I'm sure that you guys are going to play a big role in it. So thank you all for joining us, and maybe we have Paul Rothman come up and moderate some questions.
Thank you, Bill. Now you can see why I am honored to be the dean of this med school with faculty like that. Aren't they terrific? So thank you. So we're going to open the floor up for questions from these people. Anything that about what you've heard or other questions that you might ask. So I'm going to go, the question is, there's an idea about silos between different academic medical centers, and tell a little bit about sharing ideas between different scientists throughout the country.
Two good examples, but sharing cancer genetic information.
Yeah, the type of technologies that we've talked about, they really work best when many minds from across the country, not just in our institution, come together. And the way that genetic medicine has evolved over time and the way we can use these technologies has been in large part because we have broken down a lot of the types of silos that used to exist perhaps in other times, and still do exist to some extent. But I think that the tendency more and more as people that are really interested in solving these big problems are thinking about is to actually figure out how to work together to first get the technologies to make these very important, incredible measurements, but then to also work together with the right colleagues to think about how to solve the problem of how we interpret all that information. It's just such a voluminous amount of data that we can collect now that it puts things like the big data problem in a totally different context. We think about big companies like Google and things like that when we think about big data. But YouTube, as an example, they have a scale of data that's in the range of exabytes, which is 1,000 petabytes, which is 1,000,000 gigabytes a year, and that seems like a huge amount of data. With genetic sequencing alone, in the way that it's being carried out now, it's going to be somewhere around 20-fold that per year for YouTube. So that type of big data is just enormous, and if you think about it, it has to be that way. Every person has big data worth of information just within that person, and we're making those measurements across a population. So it's just an enormous undertaking that can only be done when we share the data and think about it together across any prior silos and barriers. We got to do it all together. So these barriers that people would think about and be intimidated by before, we're trying to figure out active ways to break down all those barriers and really work together on this problem.
Unstructured.
Well, and I would just add on that. This is where an entity like the Cancer Center really shows its value in beyond the treatment of disease directly. The Cancer Center has programs with clinical Bill, how many departments are in the cancer? I mean, there's, probably 20.
35 departments.
35
across
departments.
5 schools
But we're
and universities.
intermingled into various groups of overlapping interests, and we meet regularly and we share ideas, and that's what really allows us to try to get past the silo effect, which, as you point out, can be a real problem, a real barrier to discovery.
We have a question right here. Go. I can repeat it if, yeah. So the question is immunotherapy, which is a brand new type of cancer that we talked about, that David talked about, that helped with his lung cancer. What cancers is it effective in, and which ones have we not?
So the current wave of immunotherapy you're hearing about are the use of what we call immune checkpoint inhibitors. And they appear to work the best when all the immune cells that might otherwise kill cancer cells have all gone to where the cancer cells are and they're ready to get them. But what's happening is, those of you who raised adolescent daughters will feel with me on this one. They're getting the talk to the hand response from the cancer, right? And so what these drugs do is they pull down the talk to the hand response, unleashing these immune cells to kill the cancer. So when that condition is present, these drugs work extremely well, which is half of melanoma, 15-20% of lung cancers, many other diseases. The problem for many of the rest of them is that there aren't the immune cells in the cancer yet. One of the things that Mr. Gobin did is one of the earlier clinical trials he participated in, and he knows the jargon drug name for it, 5-AZA he'll call it, azacitidine. We noticed that a disproportionate number of people who participated in that trial, got azacitidine with lung cancer, ultimately responded to nivolumab, which is the name of the drug, that brand name's Opdivo. And that turns out to probably not have been an accident. By having discovery researchers go back and say, why is that? Why did that work? We now realize that when lung cancer is treated with azacitidine, they don't shrink and go away. As he told you, that sort of chilled out a bit, but they start to look like they've been infected by a virus. And they start to throw up the talk to the hand response, and immune cells show up. So can we improve how well these drugs work by some trick like azacitidine? And that's what all the research is about now. There's people that get tremendously benefited by these drugs alone. How can we extend the benefit? Cancers that don't respond very well, colon cancer, ovarian cancer, breast cancer, prostate cancer, we need to do better.
This is all very exciting. Can you tell us what's going on with anything for early detection drugs and maybe even a preventative vaccine?
So the question, for those who couldn't hear, tell us about preventative drugs and perhaps preventative vaccines.
Well, let's start with early detection, because that's one I know I can flip to Vas. And hey, you can detect DNA at the crime scene in a hair follicle and convict somebody. Can we convict a cancer?
Yeah, and actually you put it in the best way, because the same types of technologies that we've been talking about before, we can now tune in such a way that we can find very rare mutant molecules that are emblematic of how a cancer cell evolves and originates, and see if we can find that in the sea of other normal DNA that might be present in someone's blood or urine or other biospecimen. And different strategies for making those kinds of measurements, of finding the rare cancer molecules in the sea of other normal molecules, are being tested and making their way through in labs at Hopkins and elsewhere as well. And the key to that is to try to figure out how to deploy those in the right settings, in screening, in diagnostic modes, and others, to make sure that we can detect those cancers early. But to also keep in mind that as we have ways to detect cancer earlier and earlier, we also need to have tools that help us figure out which of those cancers truly need treatment. Because it's possible that some of them might be indolent. And as we find them earlier and earlier, we might really need to focus our attention on trying to figure out which of those we should definitely treat aggressively at that time, and which we might need to leave alone because, for instance, the immune system, which is surveilling the body for these types of cancer formations, might do the job on their own. So this type of technology is rapidly evolving, And as we make further and further progress in that, we'll have to keep in mind what the horizon will also bring in terms of challenges and sort of meet those head on.
And I have to say that Johns Hopkins has been the leading institution in the world driving the discovery and development of these technologies and the commercialization of it. And I'll bet there'll be some DNA detection technologies used for screening and early detection in the fairly near future, within a year or two. As for vaccination, it turns out vaccines against viruses and other microbes that ultimately cause cancer are proving to be unbelievably effective. The best, more recent story is human papillomavirus vaccines. Of course, many of you are aware of this as a vaccine to try and prevent cervical cancer. But as these vaccines are being refined more and more, and one of the leading figures for this is Richard Roden at Johns Hopkins, helped build one of the original HPV vaccines that's sold by one of the larger drug companies. But he's also building the next generation that pick up more and more and more of the family of viruses that cause oral cancer and cervical cancer, so we can vaccinate hopefully the entire world effectively to rid the world of these viruses and hopefully rid the world of those problems altogether. So.
Great, thank you. One here and then one there.
Are you able to comment on Israel's cancer cure breakthrough that this article about Israel having a breakthrough of cancer?
I'm sorry, I didn't hear, I couldn't
Hear that?
hear that.
Are you able to comment on the supposed Israel's cancer cure breakthrough? It's in some article.
Cancer care breakthrough?
Yes, I can. What they did was they did a version of phage display, and what they have is an idea that oftentimes when people pick a cancer treatment against an individual target and it's effective, what we very often see is the cancer can be effectively treated and then it may roar back resistant to the treatment you selected. This instability of the genome generating many, many variants creates a substrate that's hard to defeat, even by the immune system. So what they're trying to do is, if you will, it's an old idea from a great Johns Hopkins figure, I think he was birthed it, named Donald Coffey many years ago. He's a mentor to many of us, which was to turn evolution on evolution. That's what Don Coffey used to say. And try and get a cycle time of building a new reagent that can hit the surface of a cell. As it changes, you treat it, it comes back, build a new one, build a new one, build a new one, build a new one, and that's what they're trying to do. I don't know how well it would work. Things like this have been tried. Conceptually, it's very interesting. That helps.
I've been reading a lot about biomedical engineering. Is Hopkins involved in that? And can you explain exactly what it is?
So the question is about biomedical engineering. Well, in fact, Johns Hopkins is the leader in biomedical engineering in the world. We have the #1 department. The chair of that department, Mike Miller, is Mike still here? He's here somewhere. He left. So actually, we are leaders in biomedical engineering, and that is what biomedical engineering is, bringing the fields of engineering and medicine together to tackle problems. That could be using computers to understand the brain. It could be building devices that are used in the heart or orthopedics. It could be a lot of the genome studies we do bring engineering principles. It can bring the great big data people. It can be harvesting the electronic medical record using new computer paradigms to predict people who will get sepsis or fail. And so there's a whole range of things, and we're blessed to have the best department in the world at Johns Hopkins. And Ed's less interesting. And the interesting at Hopkins, it's a department that is half in medicine and half in engineering. It's a shared department, and it's really superb. So thank you for the question.
Biomedical engineers are widely distributed among the teams trying to solve the cancer problem.
We have a question here.
I don't know how many here have a non-scientific background, but I'm amongst them. And I hear a lot of talk about gene sequencing. Now, in layman's terms, can you describe for me what a gene sequence is and its relevance?
Fasten, you want to go take that on?
Yeah, let me try to boil it down for you. So the DNA is what we're trying to understand. And that DNA, as we talked a little bit about, is a string of letters, essentially. It's like the A's, G's, C's, and T's, and they're strung together in all different orders. And what sequencing is trying to do is to read the order of those letters. So in a given sample, we're trying to figure out what is the specific order of A's, G's, C's, and T's in that sample, and try to use that readout to figure out how those samples differ from another sample. So that's really what sequencing is. It's the ability to read the DNA and to understand it.
The sequence is quite literally a code, and so it's to decode the information.
Great. We have time, I think, for one more question.
Would we be today before the genome was decoded? Seems to me to be key and integral to almost everything we're doing.
So, the Genome Project began, it took 10 years and $100 million to do the first genome. We now can do anyone's genome in this room in one day for $1,000. In fact, we now have chips that can give us all that same information that will cost about $200. So in fact, everyone in this room within the next decade will have their genomes tested. It'll be on your iPhone. You'll be able to go to a doctor and give that doctor your genome. And it'll be important in everything from understanding what drugs you can take because you metabolize them correctly, to what are your predisposition to diseases, to where in the world your parents came from or your great-great-great-grandparents came from. So there's many things we're going to learn from our genomes in the future. Given that, I wanted to thank everyone for, I'm sorry, we have one more? We could do one quickie?
Yes, you mentioned the multi-billion-dollar NIH funding of medical research, but you didn't mention the congressionally directed medical research program that is under the Department of Defense. Are you seeking and obtaining grants for that? My understanding is that whereas NIH focuses more on clinical, the Department of Defense program focuses more on basic science and research. Is Hopkins going after and receiving grants under that program?
So there are actually several federal agencies that fund research. As you said, NIH is the biggest. The Department of Defense has some programs, and we do very well in that. Actually, the people who did the penile scrotal transplant, that work's supported by the Department of Defense. There's a National Science Foundation that supports very fundamental research. Even the FDA gives out grants. So there are a variety of federal agencies. NIH is the biggest. DOD and NSF all give grants. And we across the university, not only us, School of Engineering, School of Arts and Sciences, all go after those. So thank you. Yeah. So again, thanks everyone for coming. This was a great, let's thank our speakers one more time. I want to thank everyone for taking time out of their busy days. We have a reception that's right behind us. There are doors that are going to open up to get a little food and beverage. So thanks everyone for coming again.